So the next topic we're going to study, that is immunity. So today you are feeling healthy because you have a system, some soldiers which is protecting you against the pathogenic attacks or the infectious agents, against those pathogens. Okay. So outside our body, even inside our body, there are like microbial environment, and they have virus, bacteria, even fungi, many protozoa, many multicellular pathogens as well. We call them helminths. They are trying to attack our body. So every minute, every hour, the pathogens are attacking us, but we have soldiers inside us. They are protecting us against all of them. So every day we are exposed to large number of infectious agents, and these infectious agents are called pathogens. However, only a few of these exposures result in disease. Why? This is the time that our immune system is getting failed. So it's failure of our immune system that is causing diseases, causing infections. Okay. So what is immunity? Immunity is the capability to fight against infectious agents or pathogens. Now you know that pathogens can be bacteria, virus, fungi, protozoa, or helminths. Okay. We call these infectious agents pathogens because pathos, that means pain, and gen means agent. Pain causing agents are called pathogens. Ultimately, these agents are responsible for diseases or infections. And the soldiers inside our body, they are part of our immunity. So the immunity is conferred by our immune system, right? Immunity is conferred by our immune system. So when you talk about immunity, basically there are two types of immunity. One kind of immunity that is non-specific type, that is present from our birth, and another type of immunity that is that will be developed in our body in the course of our lifespan in response to any infection or pathogenic attack. So the first one, there is innate immunity, non-specific type. The second one, the acquired immunity or adaptive immunity, that has the specificity against various pathogens, right? So when you talk about immunity, the first type of immunity, we call it innate immunity. The second type of immunity, that we call adaptive immunity or acquired immunity. Innate immunity versus acquired immunity. Okay. Innate immunity, they're present, the components of innate immunity, they're present from the time of birth, and innate immunity is non-specific type of immunity. innate immunity is non-specific type of immunity. That means the components of innate immunity, the cells of innate immunity, the soldiers, they randomly attack the pathogenic elements or pathogenic cells, right? But when you talk about acquired immunity, that is also called as adaptive immunity. That means that was not present since birth. After birth, when we interact, when the pathogens are trying to invade inside our body and we fall sick, there is infection inside our body is trying to cope up with that infection, then our body will, in response to that infection or disease, our body will prepare another kind of immunity that is more specific type, that has different attack mechanism towards different pathogenic elements or antigens, that is called acquired or adaptive immunity. So obviously that has specificity, that can be defined by specificity, that has most importantly memory. Once I am attacking you, the next time you will keep the memory of mine and you will prepare yourself to make yourself more stronger. So the subsequent attack or second attack of mine, in that case I will be lost to you because already you were prepared. So that is called memory of the primary attack, right? So it has the specificity, memory, and one more thing, it has the ability to differentiate between self and non-self. The cells which are part of my body, the cells or the components or the proteins or the foreign particles which are not part of mine. So that is called tolerance. So, acquired immunity can be defined as specificity, memory, and tolerance that is developed in response of pathogenic attack. Again, this acquired immunity can be of different type. It can be humoral immunity. It can be of cell-mediated immunity. In some other terms, there are active acquired immunity, there are passive acquired immunity. So in different terms, we can explain the topic of acquired immunity. So let's dive into detail. So first we will talk about the innate immunity, innate, that is present inside us, and since our birth, from the time of our birth, that was always present inside us. Okay, so that's why he's saying that it is non-specific immunity present at the time of birth, and it includes four types of barriers. Now, if I am a pathogen and I want to invade your body, then I have to interact with these four type of non-specific barriers. They will try to kill me if I am the pathogen. Okay, so let's talk about those four types of barriers. Okay, so the first barrier that we call physical barrier, or you can call them anatomical barrier. They're real parts of our body trying to stop acting as a shield against those pathogenic attacks. So what are they? Physical barrier or anatomical barrier? Basically, they are our skin, Skin produces lactic acid with sweat, and that makes the surface of the skin that is quite acidic in nature. Okay, and a skin also has the cuticular outer covering, the epidermis. Okay, as well as mucosa layer. Mucosa, the inner lining of the entry point, like the inner lining of gut, inner lining of respiratory tract, inner lining of urinogenital or reproductive tract. Okay, these are the entry points of the passage of those pathogens. So if we protect this inner lining of gut, respiratory tract and reproductive tract with special epithelial tissue, that is mucosa epithelium, when we are in need, they will start secreting mucus, and that alkaline mucus will sometime will kill that pathogen or will trap the bacteria or virus trying to enter inside our body. So skin and mucosa, they are physical barrier. Also, we call them first line of defense. Okay. Now, what after that? Now, the second barrier, when the bacteria ultimately will get inside our body by crossing this physical or anatomical barrier, our physical mechanism will try to stop it. So, second barrier is physical barrier. Now, what are the things comes under physical barrier? Tear. When you cry, the tear contains lysozyme, saliva. Tear and saliva, they contains antimicrobial elements called lysozyme, an enzyme which can kill bacteria. Okay, the HCl of stomach, right? Okay, then digestive enzymes, bile, then our earwax, which is called as cerumen, which is released by ceruminous gland inside our ear, that waxy material produced inside our external ear, that also has the antimicrobial property. Okay, even fever, right? Fever and urination, they also take part, like to stop the bacterial or microbial attack as physical barrier. Now, how fever works? We think that fever is a disease, but remember that fever is not a disease. Fever is a symptom of a disease. Okay, so what happens when we are infected? Our hypothalamus increases our body temperature beyond 37 degrees Celsius. Our body thinks that if I increase my body temperature, that higher temperature may kill the pathogenic agents. So that is called fever. Okay, even during the time of defecation also, we are trying to, our body is trying to eliminate few of the gut pathogens. So these are part of physiological barrier. Okay, now the third kind of barrier will be certain cells, cellular barrier. Now, particular kind of WBC, they are involved here. Not all the WBCs are part of cellular barrier. Few WBC, they are part of white blood corpuscles, they are part of cellular barrier. So what are they? Who are they? We call them PMNL, polymorphonuclear leukocytes. Yes, WBC with irregular shape of nucleus. Basically, they belongs to the group of granulocyte WBC. Okay, there are few phagocytic cells as well. There are few phagocytic cells as well, like neutrophils, macrophages, monocytes, and also another type of cell, we call them natural killer cell or NK cells. They are also part of cellular barrier. Okay. Now, if you don't understand what is phagocytosis, these macrophages and neutrophils are the primary phagocytes, and also macrophages also shows phagocytic nature. Now, what is phagocytosis? See, if you observe the amoeba, how amoeba is catching its prey. Similarly, few cells of our body will exhibit this phagocytosis movement, a movement which is produced by cytoplasmic streaming and cell membrane also help the cell to produce artificial food kind of structure that is called pseudopodia. With the help of this cytoplasmic food, they capture the prey like amoeba and digest that. So this whole process is called phagocytosis. When a cell is eating something, that process is called phagocytosis. Okay. Now I was talking about these macrophages. They are called phagocytes, even monocytes. So when these macrophages, they are present into blood or bloodstream, we call them monocytes. But when they enters into the tissue system, into the battleground, now you will find that they have enlarged pseudopodia like structure. Now we call them, these monocytes are converted into macrophages. They will kill the unicellular pathogens and even they will clear the debris where the infection is taking place. Okay. So the third barrier after cellular barrier, the fourth barrier, that is cytokine barrier. Okay. Now what do you mean by cytokine? Virus infected cells, they releases certain kind of factors, we call them cytokines. Example you can say interferons. This interferon, basically they act as stimulator. They will stimulate the neighboring cell and they will stimulate the neighboring cells. They stimulate other healthy cells and these cells will release antiviral protein to check the virus attack. Okay. Now how these cytokines are acting? This is the virus infected cells. Suppose the virus is attacking the cell. Now if I beat you, you will cry. Similarly, suppose the cell is also crying. That is a signal of the cell. And how cell will release the signal? The cell will release the signal as cytokines. If I start beating you, you will shout and you will cry, and your friends will come to save you. Similarly, when the cell is attacked by virus, this was a healthy cell. Now this is under infection of this particular virus. The cell is secreting cytokines. Now this cytokine will give signal to neighboring cells, and these neighboring cells, they will start uh, producing antiviral proteins. So that's how cytokine barriers works. Okay, so this physical barrier, cellular barrier, and cytokine barrier, they act as second line of defense. And they're part of innate immunity. They're part of innate immunity as well. Now, if we talk about the third line of defense, then we have to learn about acquired immunity or adaptive immunity. Acquired immunity and the specialized lymphocyte cells, T lymphocytes, B lymphocytes, and antibodies produced against the pathogenic toxins or antigens. Together, these are the components of acquired immunity and establishing the third line of defense of our body. Right. The same thing is written in NCERT. So NCERT is saying, when you talk about the first barrier, that is physical barriers, they prevents the entry of microbes, that is skin or mucus coating. And where this mucus coating is present, inner lining of respiratory tract, gastrointestinal tract or gut, okay, alimentary canal, and urinogenital tract. Now the secretion of mucus traps the microbes. Okay. The second barrier, physiological barrier, they prevent the microbial growth. How they are preventing? Secretion of gastric HCl. If with food, if you are intaking some pathogens, then the HCl will kill them. Even the saliva and tear, I said they have the lysozyme that has antimicrobial property. Lysozyme is a special kind of antimicrobial enzyme. Okay. Third barrier, the cellular barrier. We have here phagocytic cells. Phagocytes means neutrophil, macrophage, and monocytes. Few WBCs like polymorphonuclear WBCs or PMLL because of the specific shape of their nucleus. Okay, like neutrophils, okay, monocytes, also natural killer cells. This kind of lymphocytes also part of cellular barrier, also macrophages. Okay, the fourth barrier, cytokine barriers. So virus infected cells secrete cytokines protein called interferons. You need to remember the example of cytokines, interferons. It protects the non-infected cells from further viral infection. Okay, how? By stimulating the non-infected cells to release antiviral protein. Okay, now let's talk about acquired immunity. Acquired immunity is also called as adaptive immunity. Okay, It's pathogen specific. It's called specificity of this particular immunity, and developed during lifetime in response to infection. Right. It is characterized by memory. Now, what do you mean by memory? During the first encounter of a pathogen, body produces primary response that is of low intensity, and the second encounter or subsequent encounter of the same pathogen causes a secondary or anamnestic response that is of high intensity. So let me elaborate this with a graph. So this axis or x-axis is axis of time or pathogenic exposure, and y-axis is the axis of uh, intensity of immunity. Intensity of production of antibodies. What do you mean by intensity of your immune response? The more number of antibodies are produced against the pathogenic attack, that is the immune response of high intensity. Less the number of antibodies are produced inside our body, that is a response of low intensity. Okay. So the graph will be like this. Suppose this is the point of first infection or first exposure of a pathogen called X pathogen, the first infection. Now, in order to cope up with this infection, our body will produce this immune response, this point A, that can be called as primary response. And during the time of this primary response, mainly our body will produce the antibodies called IgM, the predominant antibodies produced during the time of primary response, that is IgM type. Okay. And this primary response is kind of a slow response. Our body will take time to produce the immune response against this pathogenic attack, and it is of low intensity. See, very less number of antibodies are produced against the pathogenic exposure or against the anti gen. Okay. Now, what after this attack? After this attack, our body will produce some memory cells. Special types of B cells are called memory cells. Now, once these memory cells are produced after the first response, after the primary response, because these memory cells will be preserving all the data which our body has observed during the time of this first pathogenic infection. So, by utilizing this data, these memory cells will produce more number of antibodies upon the next attack or subsequent attack. Okay. Now, this is the place where the second exposure took place, second exposure of that same pathogen, that is X pathogen. That can be a particular virus or particular bacteria. Okay. So upon the second exposure, very rapidly our body will start producing a particular antibody called IgG, and also very less number of IgM. So predominant antibody which produced during the time of the secondary response at that point of B, this is secondary response, that is IgG type. Right. And this secondary response, generally the secondary response is very rapid, very quick, and the secondary response is of high intensity. So this is the graphical representation of the memory power of our acquired immunity. This is called primary response, this is called secondary response. The secondary response got another name. Secondary response is also called as an amnestic response. Now what do you mean by amnestic? Amnesia means if you're forgetting everything, so that condition is called amnesia. Anamnestic, the opposite term of amnesia. Anamnestic means your memory power is sharp, you can remember the previous things, the previous attack. So that's why the secondary response, basically based upon the principle of the memory, even based upon this principle of the memory power of acquired immunity, will be developing vaccines and immunization techniques, right? Okay. What else NCERT is saying? This primary and secondary immune responses are carried out with B lymphocytes and T lymphocytes. Now we have to talk about the components of acquired immune system. Basically the cells involved, they are certain kind of lymphocytes, the smallest WBCs of our body, B cells and T cells. Okay. When you talk about B lymphocytes, they're responsible for production of antibodies. These are the proteins to fight the pathogens. Okay, these are the proteins which are soluble in nature, soluble in plasma of our blood. Okay, and when you talk about T lymphocytes, basically they help B cells to produce antibodies. So let's elaborate B cell and T cell. First, I'm going to talk about T lymphocytes or T cells. So when I'm talking about T cells, they are produced inside the primary lymphoid organ called bone marrow, but they mature inside a particular endocrine gland called thymus, which is present near to our heart. So their matured site of maturation is thymus gland. That's why we call this thymus gland as the training school of T lymphocyte. Okay. Now from this first letter of thymus, the name thymus gland, T, we have kept the name of this lymphocyte as T lymphocyte, is because the site of maturation is thymus. T for thymus, T for T lymphocytes or T cells. Okay. If I am talking about their types, the T cells can be of different types. T helper cells, most important T cells, T killer cells or cytotoxic T cells. Okay. Uh, T memory cells and tumor suppressor T cells. So they are these are different types of T cells produced inside our body. Okay. And the immunity which is exerted by these T cells, that is called cell mediated immunity. Also we call it CMI. What happens basically, these T cells, they directly interact with pathogenic cells, and they releases some factors which can either stimulate B cells for their maturation. T cells, they are either responsible for B cell maturation, or they releases certain kind of antimicrobial factors or antimicrobial proteins. This directly kills the pathogen, right? So T helper cell, they have different function, they are responsible for different function. T killer cells, they have different function. T memory cells, they're responsible for other work, and tumor suppressor cells, obviously they're responsible for checking the uncontrolled growth of cells inside our body, right? And this cell to cell, directly these cells are responsible to develop our acquired immunity, and these are the cells which are responsible for graft rejection. Okay, now what do you mean by graft rejections? Upon organ transplantation, the new organs or the new tissue which we have taken, the patient has taken from other human being, so that particular organ or tissue is called graft. Now, prior organ transplantation, you need to check the blood groups, you need to check the match of tissue match of human leukocytic antigens, HLA. So there are n number of tests to be performed in order to match the tissue type of the patient, that is the receiver or recipient or the donor of the tissue. And if the tissue are not matched properly, then the T cells will not recognize the new transplant or new graft, and it will create an immune response, and that may kill the newly transplanted organ. So that is called graft rejection. So graft rejection is performed by T cells. Remember it. Okay. Now we'll talk about B cells or B lymphocytes. Site of production and maturation is bone marrow. Production of B cells takes place inside the bone marrow, as well as maturation also takes place inside the bone marrow. Now, what is the reason behind the naming of B cells? Is because their site of maturation is bone marrow? No. For the first time, the B cell were discovered into birds, a lymphoid organ of bird, that is called bursa fabricius. Bursa fabricius in birds. So, as because the site of maturation in birds, that is bursa, B, we got this letter B from that particular word. Okay. What else about B cells? Upon maturation, they're responsible for production of antibodies. They produce what? Antibodies. They're responsible for production of antibodies. Okay. How it happens? When for the first time, if you observe a B cell, this is called immature B cell. Immature B lymphocyte. Okay. On maturation, it get converted into plasma cell. The matured B cells are called plasma cells, and this plasma cell, they are responsible for making soluble proteins in plasma, which are responsible to check the attack of antigens or the pathogenic toxins. So these structures are called antibodies. So antibodies are produced by plasma cells. They are basically matured B cells, and these antibodies are released into plasma of blood. So as because this plasma that is acting as body fluid, and the body fluid is also called as humor. So the immunity exerted by B cells, that we called humoral immunity. B cells are responsible for production of antibodies, and antibodies are released into plasma, that is a body fluid or humor. So that's why we call this immunity as humoral immunity. Also, you can call it antibody mediated immunity. Antibody mediated immunity is also called as AMI. Okay, now it's time we'll talk about antibodies. So antibodies, basically they are soluble proteins. They're also called as gamma globulins. There are different types of globulin proteins present into our plasma: alpha, beta, gamma. The gamma globulins, they're basically antibodies. Also, antibodies are called as immunoglobulins. They are also called as immunoglobulins. Right? Okay. Now, B cells will be producing different types of antibody in response to different types of antigens or pathogenic attack. That is called the specificity of acquired immunity. Right? Okay. Now, if I am talking about the types of antibodies, remember the term damage. Now, pick the letters from the word damage. Now, as because we call them immunoglobulins, they can be expressed as Ig in shortcut term. So, first it's IgD, first word IgD, then second first letter IgD, second letter IgA. If we take the third letter, that is IgM. If we take the this letter G, this letter IgG, the last letter IgE. So the letters present in the word damage: DAMGE, IgD, IgA, IgM, IgG, IgE. These are the 5 different types of the major types of antibodies. Now let's take some information about them, important informations. So IgD, they are present on B cell surface. They help in the process of interaction of B cells in the course of maturation. So B cell surface antibodies, they are IgD, IgA. Basically, they are present, these IgA, these are antibodies present into body fluids, present into mother milk, present into saliva, present into first mother milk called colostrum, saliva, even present in mucus. When you talk about IgM, they are the largest of antibodies. From the word macro, we got this term M behind the name of IgM. Okay, they are also pentamer in nature. IgM, they are pentamer in nature. This is how IgM looks like. The pentameric molecule, the largest antibody. Okay, the presence of IgM, can you remember the mechanism of the memory action of acquired immunity? After the first exposure, the most predominant antibodies, they are IgM type, right? So the presence of IgM denotes fresh infection. If I talk about IgG, they are the smallest among antibodies and they are most abundant antibodies present into our body. More the numbers of antibodies you will be finding into our body, they belong to the group IgG. And one more important thing, they can cross, is because they are the smallest antibodies, they can cross placental barrier. They can cross placental barrier. That's why when in the chapter of body fluid and circulation, you will learn about a disease called erythroblastosis fetalis, like hemolytic disease in newborn, right? That is because of this particular type of antibodies called Rh antibody, which will enter from mother's blood to fetus body and will attack the RBC and the liver of the fetus, and that will cause the erythroblastosis fetalis into that growing child inside the mother's womb, is because these IgG kind of antibodies, which we are calling Rh antibodies, they are killing the matured RBC inside that fetus and leaving the immature RBCs called erythroblast. So that's why we call the disease called erythroblastosis fetalis. So when you will talk about that disease erythroblastosis fetalis, you have to remember the name of IgG antibody. The smallest antibody, they can cross the placental barrier. Okay. Now IgE, they're the antibodies responsible for allergic reaction. Okay, so IgE, they are responsible for allergic reaction. Whenever there will be allergy into our body, that means our body is producing more number of this particular antigen, antibody against some allergens or antigens, and the antibody are produced inside our body, that is IgE. Okay, one more important thing about IgA. See, if someone asks you about blood grouping antibodies, mark it properly, focus it properly. We know that if we follow the ABO system of blood grouping, we have like A blood group, B blood group, AB and O blood group, right? And also you have A positive, B positive, A negative, B negative, ABO positive, ABO negative, O positive, O negative. So we know this blood grouping is done on the based upon the presence of antigen on the surface of RBC. Blood grouping is done on the like based upon the presence of antigens on the RBC surface, right? But when you study these blood groups, we also study that there are certain antibodies present into our plasma. Like if a person is having blood group A, he or she must have the A antigen on RBC, but anti-B antibodies in plasma. If a person has blood group B, that person has B antigen on the surface of RBC, but anti-A antibody in plasma. If a person has blood group AB, if a person has blood group AB, both the antigen A and B are present on the surface of RBC, none of the antibodies are present into the plasma. If a person with blood group O, he or she has no antigens present on the RBC, but both the antibodies anti-N and anti-B present in the plasma. So how these antibodies are produced? Are they present during the time of birth? No. These blood grouping antibodies, remember, they are examples of acquired immunity. Blood grouping antibodies are example of acquired immunity. Upon subsequent exposure of multiple bacteria or pathogens, our body will develop those anti-A or anti-B antibodies. They are not present during the time of birth. That's why we say the reliable method to determine blood group is not antibody present into our plasma, but antigen present on the surface of RBC. And those blood grouping antibodies are basically IgA type. If we talk about anti-A or anti-B, they are IgA type, but anti-Rh or Rh antibody, they are IgG type. So that was all about different types of antibodies. Now I will talk about the structure of a basic antibody molecule. Okay. Now when you talk about the structure of basic antibody molecule, we express is like that H2L2. Basically, the structure of antibody molecule that is represented as quaternary structure of protein, made up of four subunits, four polypeptide chains, two are heavy chains, so H2, two are light chains called L2. So that's why structure of antibody is represented as H2L2. Now let's draw this structure of antibody. These are the four chains, polypeptide chains, and they are connected by disulfide bonds. Sulfur-sulfur bonds are called disulfide bonds. So in this structure of antibody, whatever the structure, we are going to find two heavy chains, two light chains, 16 disulfide bonds, sulfur-sulfur bonds or disulfide bonds. How many? 16 disulfide bonds. Okay. Antibody molecule also has a variable region and a constant region. Remember this. So how many disulfide bonds are there? 16. So total 16 disulfide bonds are there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay. And if somebody asks you that how many intrachain and how many interchain bonds are there? Okay. So out of the 16 disulfide bonds, interchain bonds, total 1, 2, 3, 4 interchain bonds are there. So 12 intrachain bonds and 4 interchain bonds are present into the structure of antibody molecules. Okay. These two are the light chain marking it L1 and L2, and these two are the heavy chains marking them H1 and H2. Okay. And this part is called the variable part, and this part is called the constant part. Okay. Now if we consider each of the polypeptide chain, we'll find that here there is presence of N terminal, and towards the end of constant part, there is presence of C terminal. Why it's so? If you check the primary structure of protein, they're basically made up of polymerization of amino acid. So the first amino acid that has undisturbed amino group, that is called N terminal, and the last amino acid that has undisturbed carboxylic group, that we call C terminal. Okay, so the variable region has the N terminal and the constant region has the C terminal. Again, one more important thing, towards the variable region, we'll find the antigen binding sites. This is antibody, so antigens are gonna bind with this structure. So antigen binding sites, so if this is the antigenic molecule, this is gonna bind with the antigen binding site present on the variable zone. This is the antigen. This is binding with the antigen binding site. So antigen binding site present into the variable region. Okay. So that was all about the structure of antibody molecule. Now let's move forward. The types of acquired immunity, that is active immunity and passive immunity. Okay, before jumping into the active and passive immunity, let's talk about one more thing, that is the tolerance of acquired immunity. Tolerance means our acquired immunity, specifically the T lymphocytes, they can differentiate between self and non-self cells or antigens. So that property is called tolerance. And you need to maintain your tolerance of your body. If the tolerance is failed, then we'll get a disease called autoimmune disorder. We'll get a disorder called autoimmune disorder. Okay. So how this tolerance is created? So basically, so immature T cells, they are called naive T cells. They are proliferated and matured into thymus, right? Now when this maturation process is going on, few of the like T cells will modify themselves. There will be mutation inside few of the T cells. So eventually what will happen, if the T cells successfully can produce its own colony, then these modified T cells, they will try to attack our own cells, right? So what our body does, if our body finds that there is abnormal T cells are produced, then and then our body eliminates that. So that process is called clonal selection, right? Only our body will pass or give permission for the proliferation and differentiation of the normal T cells, which will never attack our own body cells. So this is the process how a body will produce the tolerance and stop the process, stop the mechanism how the T cell have, or the probabilities, will destroy all the probabilities that the abnormal T cells in future can attack our own cells. So if by chance the tolerance is failed, I said we'll get a disorder called autoimmune disorder. Okay, and this is due to the tolerance only. The T cells will recognize only our own cells and antibodies and will initiate immune response or immune attack against the foreign pathogens or against the foreign bodies or foreign cells, or even this is the tolerance which is responsible for sometime graft rejection. Which cells are responsible? T cells. Okay, now let's talk about active immunity and passive immunity. Active immunity. This is the immunity in which antibodies are produced in a host body. When the host is exposed to antigens, either living or dead microbes, or the proteins of those pathogens or microbes, it's a slow process. It is produced by two ways. So what is the basic difference between active immunity and passive immunity? If this is you, upon the entry of pathogen, your body is producing antibodies against that actively by utilizing your B cells. So that is called active immunity. And you just learned that that is a very slow process, but sometime what happens, suppose a patient of snake bite or tetanus patient, so you don't have that time span to wait that your body will produce antibody to neutralize the effect of that antigen, so you need quick response. So in that case what we will do, we need to borrow antigens, we need to borrow antibodies from other human or from other animal. So the antibodies from other human produced inside their body or other animal can be inserted or injected into our body. So that is called passive immunity. So in passive immunity, you don't have to produce antibodies by yourself. You just need to borrow, or just need to take the antibodies which are produced by other man, other human, or other animals, and you just have to inject it inside your body with the help of serum. Okay. So I'm talking about active immunity. There are two types: natural active immunity and artificial active immunity. What is the difference between natural and artificial? So natural active immunity is developed during the time of natural infection by microbes, because when bacteria or virus are attacking you, suppose you got sickness, okay, you got typhoid fever, you got influenza, common flu, common cold, rhinitis, you got any kind of disease, okay. So when you are falling ill, by default your body will start producing specific type of antibodies against specific type of antigens. That is natural active immunity. But what is artificial active immunity? It is developed by injecting microbes deliberately during the time of immunization. Here I am talking about the process vaccination. So either the vaccine is injected or taken through oral drops. You think that you are taking some medicines or antibiotics? No, never. The vaccines are basically either alive or dead, or some DNA RNA segments of that particular pathogenic element. So vaccine is basically pathogen. You are taking pathogen inside your body. Intentionally you are injecting them or you are administrating them inside your body, so that they will go inside and they will threat your immunity. That is a kind of false threat, and your immunity will start producing antibodies against that real pathogen. Okay. Now if I am talking about passive immunity, here the ready-made antibodies are directly given to the body. That is also of two types: natural passive immunity, that is antibodies like IgG from mother to placenta to fetus, antibodies like IgA that is taken by the infants during the time of breastfeeding, okay. And if I am talking about artificial passive immunity, example is anti-tetanus serum or anti-venin serum against snake bite. Okay. Now let's solve this question, assertion and reason. Now this assertion is saying that active immunity is slow. Yes, obviously it's slow process and takes time to give the full effective response. That is absolutely true sentence. Reason is saying that injecting microbes intentionally during the immunization or infectious organism gaining access into our body during the natural infection induces active immunity. Yes, that is also true. The first thing, intentionally during immunization, that is the artificial active immunity, and gaining access into the body during natural infection, that is the natural active immunity. Okay, so both the assertion and reason, they are true, but reason is not explaining the assertion. That will be the answer. Okay. So in order to get the proper reason, you need to explain why this active immunity is slow and take a time for the to show the full effective response. Okay. Next assertion, they're saying that acquired immunity is pathogen specific and involves memory. Acquired immunity, that is absolutely true, and the reason said that the body produces a primary response upon the first exposure to a pathogen. Yes, exactly. Which is followed by a stronger secondary response upon subsequent encounter. So if we say like that, acquired immunity is pathogen specific and involves memory, because the body produces primary response, pathogen specific and involves memory, because the body produces primary response upon first exposure to pathogen, which is followed by a stronger secondary response upon subsequent encounters. Yes, that is basically explaining the memory part. So it is said that it can be answered like this: reason, yes, reason is explaining the assertion. Okay. Now we'll study about vaccination and immunization. See, vaccination is always active mechanism, active immunity. But if you are talking about immunization, that is a broad spectrum terminology. Immunization, it can be active, it can be passive. Okay. The administration of anti-tetanus, the administration of antivenin, okay, they are types of passive immunization. And vaccination, that is also type of immunization, that is a type of active immunization. So immunization is a broad spectrum terminology. Under immunization, we study about vaccination. Now, do you know who is father of immunization? Edward Jenner. Dr. Edward Jenner is called as father of immunization, because for the first time he discovered, he prepared the vaccine against the disease called smallpox. Nowadays we are getting the pox that is called chicken pox. There is a more dangerous version of chicken pox in 1800, in the like uh, uh, 1800 or early 1900. The disease, we call that uh, smallpox. Okay, that was a fatal disease, and after the like discovery of this vaccine against smallpox, that disease was eradicated, right? There is no existence of smallpox nowadays. Okay, so why we named this process of immunization as vaccination, the active immunization process as vaccination? Because Dr. Edward Jenner, somehow he got to know that the cowpox is related, the cowpox has the ability to produce a mild infection in human body, and if I am infected with cowpox, I will gain the ability to produce the antibodies to cope with the pox virus or the antigens produced by the pox virus, and again the subsequent infection of any other pox virus takes place inside my body is prepared with antibodies to fight against the smallpox or chickenpox. So basically we got the terminology vaccine from the word vaka, that means cow. So the cow is involved, that's why the term is called vaccination. Remember, The preparation of first smallpox vaccine was done by observing how cowpox is affecting us and preparing us for subsequent pox virus attack. That's why. Okay. So this is based upon the memory. The principle of vaccination is the memory of immune system. You need to remember that. That is the principle of vaccination. Let's talk about active immunization, that is also called vaccination. So in this, a preparation of vaccine that can be antigenic proteins or pathogen or inactivated pathogen is introduced into the body. It results in the development of antibodies. During actual infection, the antibodies neutralize antigens. The vaccination also generate memory B cells and T cells. So they recognize the pathogen quickly. That is, examples are given: polio vaccine, hepatitis B vaccine, DPT, diphtheria, pertussis, tetanus vaccine. There are other vaccines called MMR vaccine, measles, mumps, rubella vaccine, BCG vaccine, bacillus calmette guarin vaccine. Polio vaccine is also called as OPV, oral polio vaccine. Okay. Vaccines are produced using DNA recombinant technology. That is, one example is given. Recombinant DNA vaccine, that is a type of second generation vaccine. Hepatitis B vaccine is produced from yeast. Now let's talk about different kinds of vaccine. So if we talk about the first generation vaccine, first gen vaccine, they used to use dead pathogen or attenuated live pathogen. Attenuated means weakened pathogen. Okay. So these are called first generation vaccine. So what is the basic principle of vaccination? Let me elaborate into a simpler way. Suppose you are going to visit a jungle and somebody told you there you may interact with some wild animals, so take proper precaution against them. Okay, now you don't have any idea about wild animals. Now somebody is there to show you some videos or photos of lions and tigers. Now when you observe, when you watch the videos of lions and tigers, now imagine in your imagination, into your mind, you will make their, will build their image. Yes, this kind of animals are present into that jungle. So whenever I will observe them, whenever I will see them, I will hide, right? I will not go in front of them because they will kill and eat me. A similar way, our body does the same thing. Your body doesn't have any idea about the foreign pathogen. So if the first time if someone is attacking you, your body will take much time to like produce antibodies against those antigens, right? And it will take time for recovery. So what we are doing, artificially we are giving signal to our immune system, yes, this kind of pathogens are present in this environment, so please observe them and produce memory cells. So when the real actual time or actual attack takes place, you are already prepared. Quickly you may produce the secondary response. So the response of vaccination upon the real attack of pathogen, basically that is the secondary response, and the primary response will be produced inside your body just after the insertion or administration of these vaccines in form of dead pathogen or attenuated weakened live pathogen. So these are called first generation vaccine. Now if I talk about the second generation vaccine, basically they are called recombinant DNA vaccine. Here we use rDNA technology recombinant DNA technology. So it said that the vaccine of hepatitis V is produced by yeast. So yeast is acting as a host in our laboratory. So the particular genome, a particular gene we have taken from the hepatitis B virus, right? So the genome is incorporated into the genome of yeast, and now yeast will produce the proteins which are produced by hepatitis B virus. Now, if you inject this protein or administrate these proteins inside the body of a healthy person, so that will give a threat, a mild threat to our immune system, and that will produce the memory cells, right? That is acting as a vaccine. The third generation vaccine, if you talk about third generation vaccine, there the DNA vaccine or RNA vaccine. Directly pathogenic DNA and RNA are inserted into our body, and those pathogenic DNA and RNA will stimulate our immune response. Okay. Now, what is passive immunization? Passive immunization means it is the direct injection of preformed antibodies or ready-made antibodies, either we are getting from other human, or you are preparing inside the body of horse or rabbits or other animals, or antidotes or antitoxin. It is required for quick immune response. Immunization against tetanus or snake venom, right? So when a snake is biting you, the poison, not the poison, we call it snake venom. The venom may affect your muscle, the venom may attack your RBC, destroy your RBC, the venom may attack your nervous system. So different snakes have different types of venom. So we have prepared the anti-venin serum by injecting very low intensity or very low volume of this snake venom into the body of horse and rabbit, and when they are tolerant against the fetal amount or fetal volume of that particular snake venom, basically their body produces effective antibodies to stop the attack of that particular antigen present in the snake venom. So what we will do, we will collect their blood plasma, we will filter it, we will take the serum and we will inject into the patient which is bitten by snake. Okay. Now let's talk about allergies. Okay. Again, acquired immunity is involved into this. Allergy. So what is allergy basically? Every day we are interacting with numbers of foreign particles. Okay. We can call those foreign particles as harmless antigens, right? So suppose a particular flower or the pollen grain of that flower, whenever we are taking the smell of a rose flower or a marigold flower, that is not harming us, that is not producing immune response inside our body, because that is a harmless antigen. Our immune system is acquainted with it. But there is a person, there is a particular person, when he or she is inhaling that pollen grain of that marigold flower or the rose flower, that pollen grain is initiating or provoking the immune system of that particular person to produce the antibodies against that particular harmless antigen, and ultimately that is causing inflammation into his or her body. So that particular exaggerated, which is not required, which is not observed in some normal person, but observed in some particular specific group of person. So we call that exaggerated reaction of immune system, that is called allergic reaction. Okay. So allergic reaction is basically exaggerated response of our immune system against certain antigens, and remember these certain antigens, these are harmless for most of us, present into the environment. Okay, so we call those certain harmless antigens as allergens. So substances causing allergy are called allergens. There is mites present into the dust. They are minute arthropods, pollens of flowers, animal denders. If you have pet, if you have cat or a dog, then the dander may affect you. Even fur of animals, even few food materials, few have allergy against egg, few have allergy against brinjal. Antibodies produced against these allergens are IgE type, remember it. This IgE binds on the mast cells to release chemicals like histamine and serotonin for them. It results in allergic reaction. Now, what are the symptoms of allergic reaction? The symptoms will be like primarily there will be sneezing, watery eyes, runny nose, difficulty in breathing, wheezing, skin rashes, etc. Okay. Now, determination of causes of allergy. How to determine if you visiting a doctor? How he or she will determine that you have allergy against which particular allergen? So the patient is exposed or injected with very small doses of possible allergens, and the reaction is studied. Right. Now, how to do treatment? Drugs like, so basically the mast cell or basophils of our body, they are releasing histamine and serotonin. So you need to block the effect of this histamine. This is the main villain behind the allergy. So you have to use drug like antihistamine. Either it can be uh, antihistamine, any kind of. There are many enzymes which are responsible for breakdown of histamine. Adrenaline and few steroids quickly reduces the symptoms of allergy. Okay, one example is given. Asthma is a respiratory disorder due to allergic reaction. Even it said that modern day lifestyle and protected environment provided early in life result in low immunity. So in our time we used to play cricket or we used to play football. If it's raining, even we used to play, go outside and play football into fields. But nowadays even playgrounds are not available, and the kids, they're going into schools, colleges or tuition, and then again they're sitting inside the home. They're playing mobile games. Okay, they're playing indoor games. They don't know what is social life. So you have to mingle with the nature. Until and unless you are mingling with the nature, right, your immunity will not become stronger, right? So in that case there will be more sensitivity to allergens. So many children into metro cities suffer from allergies and asthma. But if you observe the village people or the children's of village, they have stronger immunity against the natural antigens. Okay, so what happens basically, suppose you have taken a particular allergen. The allergen can be pollen, allergen can be the mites present into the dust. When you are dusting your room, sometimes you may feel after dusting, suddenly you started coughing, sneezing, runny nose will be there, watery eyes will be there. So why it's so? So because these allergens will they enter into your body will counter it with immune response. And your body start producing antibodies like IgE, the allergic antibody IgE, right? Now this IgE will go and bind with the surface of mast cells, will stimulate mast cells or basophils, and upon stimulation the mast cell and basophil, they will start secreting histamine, mainly histamine. This mast cell or basophil, mainly they releases histamine, serotonin, heparin. So mainly this histamine, that is responsible for vasodilation and bronchoconstriction. So if it's dilating the blood vessel, the diameter of blood vessel when increased, the phagocytic cell will get passage to come out of the blood vessel and attack your normal body cell, and that will cause inflammation. Right. And if it's doing bronchoconstriction, your respiratory passage, due to the inflammation effect, if your respiratory passage is blocked, then there will be problem in inhaling and exhaling, and that may cause the symptoms of asthma. Right. Okay. The next, another non-infectious disease we are talking about, autoimmunity. So already we are discussing the autoimmunity is nothing but failure of tolerance. The one of the important property of our acquired immunity. So it's written here, in higher vertebrates, memory-based acquired immunity evolved based on the ability to differentiate foreign organism from the self cells, that is called tolerance. Sometimes due to genetic or other unknown reason, you have to remember, either genetic or some unknown reason, the body attacks self cells, resulting in damage of our body. It is called autoimmune diseases. Example, rheumatoid arthritis. So what happens into rheumatoid arthritis? The rheumatoid factor, basically they are modified type of IgM, they attack joints of our body. Which kind of joints they attack? Synovial, basically they attack synovial membrane of synovial joints, and that cause inflammation into our joints. We call this rheumatoid arthritis. Okay, another example, anaphylaxis, that is a kind of shock. Okay, type 1 diabetes. So here what happens, our immune antibodies destroy our beta cells of pancreas, which are responsible for production of insulin. Another example of autoimmunity, hay fever. Another example, uh, lupus disease or systemic erytheromatous lupus. Our immune system or antibodies or modified antibodies will attack our own tissue. Okay. Another example, myasthenia gravis. In myasthenia gravis, what happens? The modified antibodies, they will attack neuromuscular junction of our body. Okay. So these are different types of autoimmune disorders. Okay. Now we'll talk about the components of immune system of our body. Okay, so what are the components? The human immune system consists of lymphoid organs, lymphoid tissues, okay, some particular cells. Basically, we need to talk about WBC, and soluble molecules, like we call them antibodies, right? So the immune system also plays an important role in allergic reaction, autoimmune diseases, and organ transplantation. Now, our topic is to learn about lymphoid organs, right? So let's talk about lymphoid organs. So what are lymphoid organs? They're the organs where origin or maturation and or proliferation of lymphocytes occur. So basically, the nave of our acquired immune system is made by B cells and T cells. We need to, like in detail, we need to study the process of their production, their migration from one place to another place, process of their maturation, and process of how they are becoming the effector cells to cope with the antigens or the foreign pathogens, how they are exhibiting the specificity of acquired immune system, the memory of acquired immune system, and the tolerance of acquired immune system. So until unless we are learning about lymphoid organs, it's not possible for us to track down the total mechanism of action of B cells and T cells, right? Okay, so lymphoid organs are of two types: primary lymphoid organs and secondary lymphoid organs. Okay, so primary lymphoid organs, remember, they are the site of production, site of maturation of T cells and B cells. So what happens here? B cells and T cells are matured in such a way inside the primary lymphoid organs that they produce some, they mature B cell and T cells, they will be exhibiting their surface receptor, so they can sense, at least sense the antigens. So in primary lymphoid organs, we produce antigen sensing lymphocytes. Now what happens into secondary lymphoid organ? Now in secondary lymphoid organ, there will be further proliferation of those lymphocytes and antigen presentation takes place. Antigen presentation means, that is the means, for exam, one part is theoretically you are studying it and another part practically you are doing it. So when these T cells or B cells, they are getting theoretical knowledge, they're inside the practical lymphocytes. When they're practically experiencing the antigens and the pathogens, so that is called antigen presentation, and upon this practical exposure of antigens, they will get converted into matured B cells or T cells. We call them effector cells. Right. So the first primary lymphoid organs, that is inside the bone marrow. Bone marrow, the site of production of B cell and T cell. B cell and T cell, immature B cell and T cell, they will be produced inside, and there will be maturation of B cells as well. But where these T cells are going to get matured, they will travel near to heart. This is a very cheaper heart, right? So near to heart there is a gland where T cells will migrate, and T cells will get proliferated and matured. This is called thymus. This is called bone marrow. So bone marrow and thymus, they are part of primary lymphoid organs. They are primary lymphoid organs. The site of production and maturation of B cells and T cells. Bone marrow, site of production and maturation of B cells. Thymus, the site of maturation of T cell. T cells though produced inside the bone marrows. Now from this thymus gland, the T cells and B cells will migrate into secondary lymphoid organs. Now secondary lymphoid organs means you have spleen, you have tonsils, you have appendix, you have malt mucosa associated lymphoid tissue, gut associated lymphoid tissue, and many other organs, right? They are called as secondary lymphoid organs. So inside secondary lymphoid organs, what happens? Antigen presentation will take place, and the T cells and B cell will become matured one. Okay, so let's read what NCERT is saying. So when we talk about the primary lymphoid organs, the organs where lymphocytes are matured and differentiated into antigen sensitive lymphocytes, right? Okay, so bone marrow and thymus. Bone marrow is the site of production of B cell and T cell. Thymus, site of maturation of T cell. Most of the B cells will mature here only inside the bone marrow. B cell maturation also take place inside the bone marrow. Okay. Now, when you talk about the secondary lymphoid organs, the organs to which the matured lymphocytes migrate from primary lymphoid organs and interact with antigens, then proliferate to become effector cells. The examples of secondary lymphoid organs are spleen, lymph nodes, tonsils, Peyer's patch, a small intestine, mucosa associated lymphoid tissue, a malt, or appendix. Yes. Now there is special emphasis on malt. Why? Because they are located within the lining of respiratory tract, digestive tract, and urinogenital tract, mucosa associated lymphoid tissue, and it consists or constitute 50% of lymphoid tissue. Very important point to remember. Very important. It constitutes 50% of all the lymphoid tissue present into our body. Right. Now, we'll try to elaborate what actually happens inside the primary and secondary lymphoid organs. Okay. So, suppose we are starting with T cells. Sorry, there is entry of a pathogen into our body. So, this is a bacteria. This is the pathogen. So, when this bacteria enters into our body, it will attack the tissue, and inside our body it will attack our normal cells. And how this cell will destroyed, the cells will be killed. This pathogen will produce antigen, right? Now this antigenic molecule will go and disturb the cellular function, and gradually the cell will die. This is the normal infection that is taking place inside our body. Now, this will give a response to our immune system. This will initiate an attack towards the immune system. Now we have certain APC, antigen presenting cells. Who are antigen presenting cell into our body? They are macrophages, they are dendritic cells mainly, and few B cells, few B lymphocytes also act as antigen presenting cells. So what these antigen presenting cells are doing? These antigen presenting cells, basically they will inject these antigens they will process this antigen inside, and they will exhibit them with the help of their surface receptors. Now they may present this antigen, and if this antigen presentation is happening inside the secondary lymphoid organs like spleen, right? So what happens inside the spleen? Spleen has special reticular tissue. They trap the pathogens and antigens, and those antigens are taken by macrophages, and macrophage will prepare those antigen and will show them towards the immature T cells and B cells. Sorry, the mature T cells and B cells, and upon antigen presentation, the T cells and B cells will get converted into effector cells. So they are showing these antigens to B cells or T cells. They also have their specific receptor to sense them. Now they have become the antigen sensing cells. Suppose this is T cell, this is B cell. Now upon antigen presentation, they will get converted into different type of effector cells. If they are B cells, they will get converted into plasma cells and they will start producing antibodies. And if they are T helper cells, they will activate other B cells to produce more and more antibodies. So this is how our immune system work. Okay. So now the next topic we are gonna study, that is one of the most dangerous infectious disease caused by a retrovirus. So there is a viral disease and also fatal one. Okay. So let's talk about HIV AIDS. So the full form of this disease AIDS, that is acquired immunodeficiency syndrome. I think you all know about syndrome. Syndrome means group of symptoms. Okay. So basically what happens in this particular syndrome, a virus called human immunodeficiency virus, the name of the virus is HIV virus. So human immunodeficiency virus that attacks our immune system and they deliberately destroys the nave of acquired immune system, that is T helper cells, right? And if the T helper cells are not working, just in the previous slide I said that is the T helper cells which are responsible for waking up the sleeping B cells or other sleeping cytotoxic T cells, right? So if there is no T helper cell, the signal will not become green for few of the B cells and few other immune cells, and our immune system will remain into sleeping mode and our body will fail to respond against the attack of some other pathogenic agents, right? So the duty of, see, in one line if I say what is HIV AIDS, human immunodeficiency virus, the name itself says the immunodeficiency virus. It will kill the immune system, it will destroy the nave of our immune system, and if there is no immune system into our body, the opportunistic pathogen will attack us. Okay, but in normal day, if we have those immune system, those immune cells, they don't have the dare to attack our or to challenge our immune system. But in this particular time when we are lacking with our T helper cells, so those opportunistic pathogen will destroy our body and will be causing death. Okay. The first recorded phenomena for HIV AIDS was seen in 1981, and in human population this particular disease, it was transmitted from the rhesus monkey. You need to remember this. Okay. And this particular devastating disease, infectious disease, killed more than 25 million people over last 25 years according to the data of NCERT, right? More than the data is how many people are killed, 25 million people are killed within a span of how many how many years, 25 years. So it's quite easy to remember, 25 years has killed 25 million people. Okay. The disease is characterized by decreased number of T cells. So what is the normal number of T cells? Normal, the number count of T cells is 500 to 1500 T helper cells per cubic millimeter blood or cubic deciliter blood. During the time of initial infection, the count will fall below 500. The count will fall below 500 per cubic millimeter blood, and in case of critical case, the count will fall below 200 T helper cells per deciliter or per cubic millimeter blood. Remember it. Okay. So the pathogen, the disease causing agent is HIV. The full form is human immunodeficiency virus. It's attacking our immunity. Okay, this belongs to the group of retrovirus. Now, what do you mean by retro? Retro, the meaning of retro is old, right? Sometimes you listen retro songs, you have retro style following nowadays. Okay, so retro means, why we are calling this virus as retrovirus? They're converting their single stranded RNA virus. So they will convert RNA to DNA by the process of reverse transcription. So what happens normally into our body? If you observe the central dogma, there is DNA to DNA, that is called DNA replication. DNA to formation of messenger RNA, that is called transcription. And from messenger RNA to protein, that is called translation. Right. But if you observe the this particular virus, here there will be conversion of RNA to DNA by the process of reverse transcription. So that is following a primitive pathway. So that's why we are naming this virus as retrovirus, and obviously that is enveloped virus that has protein capsid, protein covering, and also a lipid covering enclosing RNA genome, right? So the virus contains two identical single-stranded RNA, single-stranded RNA or ssRNA we call it. Two, you need to remember the number, two single-stranded RNA, two reverse transcriptase enzyme that is responsible for converting the viral RNA into viral DNA, two integrase enzyme that is responsible for incorporating the viral DNA within the host DNA, that is acting as a paste, and two protease enzyme. These are the main components you will find inside the virus called HIV. Okay. So this is the diagram of HIV virus. So these two are called RNA core, single stranded RNA, we call it RNA core. It has integrase enzyme, it has reverse transcriptase enzyme, these two are the most important enzyme needed for viral replication inside the host body, reverse transcriptase and integrase. These are proteases. Okay. And outside that, there will be covering of protein. We call this protein covering as capsid. There will be inner capsid and there will be one outer capsid. Okay. And the outermost envelope, which is made by phospholipid bilayer. The outermost envelope that is made by phospholipid bilayer, and there are certain important spikes are embedded into this layer. These are basically receptor proteins called GP120, GP41, right? And they are acting as a spike, and basically these are the spikes which are responsible. They will attack some specific cells which have CD4 cluster differentiation factor type 4. So they are receptor, they recognize CD4 cells, right? So this virus, this particular virus, this will attack only to those cells which are displaying CD4 receptor on their cell surface. Okay, so that's why it's written here. HIV attacks CD4 plus cells. And who are CD4 plus cells? They are T helper and human macrophages. Okay, now you need to remember this T helper cells, they are the primary target for HIV. HIV also target macrophage, but HIV will not kill macrophages. Rather, HIV will like use or utilize these macrophages of our body as HIV factory. So that is called Trojan horse effect. Okay. These macrophages, they will display Trojan horse effect. So if you are good at Greek history, then you know what is Trojan horse. Okay. So this HIV kills T cells, but they will not kill macrophages, as because they will utilize macrophage as their factory. Okay. Simultaneously, when they will enter into our body, first they will show a few days of incubation period. Simultaneously, they attacks T helper cells and macrophages, and by escaping our immunity inside those macrophages, they will not create any threat against those macrophage. So silently inside those macrophages, they will create their viral population. Okay, and suddenly what will happen? There will be progressive decrease in the T lymphocyte, right? I said if the count of T lymphocyte will start falling below 500 T cells per deciliter blood, and there will be bouts of, according to NCAD, bouts means episodes of fever, as because gradually the foreign pathogens, they are trying to attack you, diarrhea, and obviously there will be weight loss. That's why this is also called as slim disease. HIV AIDS is also called as slim disease. So these are the primary symptoms, right? After initial attack. Okay, now in severe case, what will happen? It's called full blown stage. Opportunistic pathogen will try to invade and attack and establish disease into your body. So the CD4 plus count, basically we are talking about T helper cell count, will decrease below 200 cell per deciliter or 200 into 10 to the power 6 cells per liter blood. Okay, so the bacteria, mycobacterium will attack us, that will cause tuberculosis. Toxoplasma will attack us, that will cause toxoplasmosis. There will be a number of fungal infection will be taking place inside our body. Even various cancer will take place into the skin and lymph nodes. There will be haphazard entropic condition going inside the body, right? And sometime upon the initial attack, the patient may not show any kind of symptoms, so that is called incubation period. And incubation period means the period between infection to appearance of symptoms. So in between, in this particular span, the patient will remain asymptomatic, but inside his or her body the T cells are establishing their population. So it can be few months, to many years. As long as 5 to 10 years the patient can remain asymptomatic. So what is incubation period? So basically this is a healthy person, but suddenly the virus attacked, the virus started entering into the body. So this is called infection. Now the particular day the patient can really feel from inside, yes, there is appearance of certain symptoms taking place inside the body. So that is called the duration from infection to symptom, appearance of symptoms. This time duration we call it incubation period. So what happened during this time? Basically, we have our macrophages. This this virus will come closer to macrophage, and by surface to surface recognition, this will deliver a message that I am your friend, I am not your foe, I am not your enemy. So what will happen? The macrophage will give permission that the virus will enter the RNA inside that macrophage, and escaping our immune cells, other immune system inside the macrophage, the virus will establish the population, and in course of time gradually it will increase the population, but it will not harm the macrophage. By the process of exocytosis, it will go out of the macrophage. It will not rupture or kill the macrophage, because the primary target is not macrophage. Now this virus will attack T helper cell with specific kind of receptor present on it called CD4 receptor. Now this virus will go, the virus will attack, the virus will inject the RNA core and eventually will destroy the cell. Right. So that's what I was telling. Okay. So how it's spread? It's spread through body fluids or mucosa to mucosa transfer. So it will never be transmitted by the process of simple touch or even by the process of kiss, okay, or mosquito bite. There are certain misconceptions regarding the transmission of HIV/AIDS. Do not spread through touch, or physical contact like kiss or mosquito bite. Okay, remember HIV virus, they are present inside the body fluid, or they can be transmitted if there is mucosa to mucosa connection. Okay, so let's talk about the transmission of this virus, how they're getting in becoming infectious, the mode of infection. Okay, either during the time of sexual contact, right? During the time of sexual contact, there will be release of semen or vaginal fluid. Basically, these are kind of body fluid. Also, there is mucosa to mucosa connection during the time of sexual intercourse. That is, and during the time of anal sex or vaginal sex. Okay. During the time of transfusion of blood, this can happen if you are a blood donor and you have HIV titer inside your body. Okay. By the means of sharing infected needles or infected razors, even from infected mother to fetus or infected mother to child. From infected mother to fetus inside the womb, the virus can travel through the placenta, or even after the birth, by the during the time of breastfeeding, the breast milk also acting as a body fluid, that also has a titer or the good number of HIV virus inside that, so that can get transferred into the body of that newborn baby, right? Okay, now who are the people with high risk who can get the disease or get the HIV infection easily? Okay, peoples with multiple sexual partners without knowing them properly, without any medical background check, if we are doing so. Intravenous drug abusers, like there is a group of friends and they are abusing, they are taking the narcotics and drugs, and there is only one injection or one syringe, right? And they are utilizing the same syringe to insert the drug intravenously, and one of the friend of that group already infected with HIV AIDS. So from that particular friend, the to the infected needle of that particular syringe, the virus will enter through all the friend's body, right? Or even it can transmit by repeated blood transfusion. The patients with thalassemia, the patient with sickle cell anemia, the patient with bone marrow disease, so they need repeated blood transfusion within few days, even with every week, right? So these are the people, the group of people which are at the worst or the age of high risk to get the HIV infection. Okay. Now we'll talk about how to diagnose if a person is having HIV or not. Okay, I'm advising you before the marriage, we must need to diagnose the few medical tests of our partner, right? HIV is one of them. So, what is the common screening test or diagnostic test? There is ELISA, enzyme linked immunosorbent assay. Okay, but sometime the ELISA can give false negative or false positive result. Sometime ELISA can give false negative result. Means suppose one person already suffering with HIV AIDS, but due to problem into that test kit, that kit will not like undergo the process in a proper manner, and that will show that the patient doesn't is not infected with that particular disease. This is called false negative test. Means you have the disease, but the kit is unable to diagnose it. Now, what is false positive test? You are not infected with that disease, but due to the problem into the test kit, that will show that yes, you are suffering or you are diagnosed with that particular disease. That is called false positive case. So due to this phenomena, false positive or false negative test of enzyme linked immunosorbent assay, we need to search for other confirmatory test. So ELISA is just a screening test, a preliminary screening, but if you want to confirm that one has that disease or not, then you have to go for confirmatory test. Now what happens in ELISA basically? There will be particular ELISA kit. It has particular antibodies which can which are linked with particular enzymes, right? Now if I take blood from your body or body fluid sample and put inside this test kit, so suppose you have this HIV inside your body, and this HIV affected cells, they will produce viral proteins or viral RNA, right? So these viral proteins will get attached with the receptor present into the ELISA kit, and that will stimulate the enzyme. Now in presence of chromogenic substance, the chromogenic substance will respond if there is bonding or if there is reaction happening in between the antigen and antibody present inside the kit, right? So if there is successful reaction of HIV antigen and antibody reaction is happening inside the kit, that will stimulate the enzyme to utilize the chromogenic substance, and it will change the color of the kit, and that will show a positive result. Okay. Now I was talking about the confirmatory test. Okay. Now the confirmatory test, that is basically Western blot or HIV RNA test. Okay. So in Western blot, basically what we try to find, we are trying to find the proteins produced by the HIV virus inside our body. Okay, or HIV virus, if you are infected with HIV virus, then there will be presence of HIV RNA inside your body. So HIV RNA test, or there are many HIV type assay are there. Okay. Now how to do treatment by antiretroviral drugs? Okay, treatment by antiretroviral drug. There are very few retroviral drugs are present in the market, like stabudine, zidovudine, etc. Okay. Now, if you are also taking the proper precaution after the infection of HIV virus, you are visiting to doctor, taking proper medication and drugs, it can only prolong the life of the patient, but still it cannot uh, stop you from getting die. Death will be there. So the death is inevitable. By taking antiretroviral drugs, you can prolong your lifespan for a timing of for a certain a few years or few months, but you cannot escape from the death. The death will find and kill you. That's why we are saying that the death is inevitable in this particular case. Okay. Now there is a diagram of HIV infestation into our cell is given in NCERT. So we'll study that through a proper detailed diagrammatic illustration. This is the HIV virus that is attacking a healthy cell. So the whole HIV virus will not enter into the cell, rather it will insert the RNA core. It will insert the RNA core only. So the capsid and the spikes will remain outside. It will utilize the spike to identify the CD4. This particular cell has CD4 receptor on the surface, right? So suppose this is a T helper cell. So it will inject the RNA, single stranded RNA inside the cell, two single stranded RNA, right? Now along with single stranded RNA, it will also insert the integrase enzymes and reverse transcriptase enzyme. So now this reverse transcriptase RT enzyme, reverse transcriptase enzyme will start acting on those single stranded RNA. They will convert this single stranded RNA into single stranded DNA. They will convert those RNA to DNA, right? Okay. Now the function of integrates, now the function of integrase will start here. That will incorporate the DNA molecule, the viral DNA molecule inside the host genome. Now we say that the host genome is hijacked. Host genome is hijacked now. Now the host genome will not uh, produce proteins required for the cell, but it will start producing the viral particles. And once this cell is laden with newly formed virus, the virus will rupture the cell. The virus will rupture the cell that will come out. The newly formed virus, they will rupture and come out and attack new cells. The virus will attack new cells and inside your body that continuously go on killing the T helper cell. So the population of T helper will go down, sometime even it will get lowered as low as less than 200 T helper cell in per cubic millimeter blood. Okay. So it is said that AIDS has no cure but only prevention. AIDS has no cure but only prevention. The death is inevitable. Remember this. Okay. So don't die of ignorance. Please follow the steps. These are the steps, or these are the high risky areas. If you are if you have multiple sexual partners, if you are taking intravenous drugs, or if you are prone to blood transfusion, please check it properly, check sterilized things in a proper manner. If you are like establishing sexual interaction with someone, please utilize the barrier methods, like use condoms, right? You know that condoms has the capability to check the sexually transmitted diseases. Okay, and there is an organization called NACO, National AIDS Control Organization, that is monitoring the worldwide condition of this particular syndrome, a particular disorder, a particular disease called infectious disease called HIV/AIDS. Okay. Now a question is given here: which of the following statement is incorrect? So it said that HIV is enveloped virus containing two molecules of single stranded RNA and two molecules of reverse transcriptase. Correct sentence: HIV is enveloped virus that contains two identical molecules of single stranded RNA and one molecule of reverse transcriptase. So this is our answer. HIV is enveloped retrovirus, that is also true fact. HIV attacks T helper cell, that is also true fact. Now the next topic we will be studying, that is cancer. We will try to find some answer against the non-infectious disease, the major cause of death, which is one of the non-infectious disease, that is cancer. So basically we need to talk about the oncogenic transformation of cells. Okay. So if you have a cell inside your body that will divide with time, and this particular cell, it has few important genes called tumor suppressor genes, proto oncogenes. So it follow a particular geometrical progress inside our body. It will not go beyond that. It is called Hayflick limit of cells, right? But if there is a cancerous cell into your body, or you can call it malignant cell, so that will not follow this contact inhibition, particular limit of cell division. Okay, so that will start growing abnormally, and ultimately that will create lump or mass of unwanted cells into our body. We call it neoplastic cells, and they will give invitation for the growth of blood vessels to nourish this unwanted growth or unwanted lump of masses, and this is also called malignant tumor. And sometime what will happen, these tumors, few of the malignant cells will get detached, they will take the help of blood supply and will get transported from one place to another place to establish new tumor or new population of malignant cell, and ultimately that will exhaust the nutrients of your body. That will excessive growth of cells will cause gangrene of that particular part, and ultimately that will affect the normal physiological function. They will damage a particular organ or multiple organs, and the person will die eventually. So basically when you talk about cancer, proto oncogenes are transferred into oncogenes. That is called oncogenic transformation of cells, and that condition is called cancer. That's why the study of cancer is also called as oncology. So the study of cancer is typically based upon how the normal genes, that is called proto-oncogenes, are getting uh, changed or getting mutated form of genes, that is called oncogenic transformation, or forming oncogenes, and they will cross all the rules and regulation which is stabilized by the genetic mechanism of normal cells. Okay, so let's study that. So cancer, it is the major cause of death, and remember, this is a non-infectious disease. Okay, why the disease is known as cancer? Now, if you observe these cells, these neoplastic cells, they look like crab. They have crab-like appendages, crab-like appearance. That's why from the word crab, we got this terminology cancer. Right. So it said that more than 1 million Indians suffer from cancer, and a large number die from it annually. Annually means every year, per year. So we need to study the oncogenic transformation of cells. Okay. I think you all must know him. Yes, Hulk. So when a normal human was bombarded with gamma rays, right? There was genetic mutation inside that normal human. Basically, he was a scientist, and something happened into the lab. There was exposure of gamma rays that changed the genes of this particular person, uh, Bruce Banner, and he converted into Hulk, right? So let's talk about, so though in his case this gamma ray that was responsible for formation of superhuman, but in daily life, what will happen in real life, if I expose you into the gamma ray, that will cause oncogenic transformation of your cell, means proto-oncogene will get mutated into oncogene, and that will cause cancer into your body. So let's explore all the causes of cancer. Okay, so the cancer causing agents are called carcinogens. The carcinogen can be physical carcinogen. The carcinogens can be chemical carcinogen. The carcinogens can be biological carcinogens. Okay, so one by one, let's explore. First, we'll talk about physical carcinogen. Physical carcinogens, basically, there are different types of radiations, right? Radiations are of two types: ionizing radiation and non-ionizing radiation. Ionizing radiation means the radiations are basically the flow of wavelengths, waves, right? So the radiation has so much energy, the radiation is carrying so much energy within it, so that can displace the electron from the orbitals of atom. So that's why we are calling it ionizing radiation. So ionizing radiation means which can convert atoms by eliminating electrons to ions. So those radiations are called ionizing radiation. Now, if I talk about then what do you mean by non-ionizing radiation? The waves. doesn't have that much energy to eliminate the electrons out, or to they cannot, these radiations cannot convert atom to ions. So that's why we are calling them non-ionizing. Okay, so X-ray and gamma rays, they are ionizing radiation, and ultraviolet rays, they are non-ionizing radiation. So if there is open exposure of X-ray or gamma ray, then obviously that will cause mutation of your gene. So basically what happens, so this is a normal piece of DNA. If this is affected by gamma ray, so that can change a particular nitrogen base. Suppose that will change the genetic makeup. So the genetic makeup has changed from, it was previously GATTCCAA. Upon mutation, it will become like this: GAA TCCAA. So their product will be totally different, right? Their product will be totally different. So if the product is different, that will, their effect on our body physiology and cellular function will also be different, right? So this is how ionizing and non-ionizing radiation can change the genetic makeup of a cell. Then they're responsible for causing mutation. Even these UV rays, their potential rays to cause mutation into our genetic makeup. That's why we say that it's ozone layer that is protecting us against these UV rays which are coming from the uh, universe, right? From the space, the cosmic rays, right? Now let's talk about chemical carcinogens. Most of the chemical carcinogens which we inhale every day in daily life, they're present into the smoke of tobacco, they're present in the smoke of industrial exhaust, even the vehicle exhaust. So if I talk about the tobacco smokes, basically they are rich in polycyclic aromatic hydrocarbons, or PAH. These are the main carcinogen present into the tobacco smokes. Nicotine is not the carcinogenic element, right? Okay. And other aromatic amines, nitrosamines, and benzene also present inside the tobacco smoke. These are the causative agent of cancer. They can cause mutation inside our body. Okay. Now, if I'm talking about biological carcinogenic agents, okay, there are two types: V oncogene and C oncogene. Okay. Now, V oncogene means oncogenic viruses. There are few viruses which will attack us, and they have a particular piece of gene inside them, and when their gene is integrated with host genome, that will mutate, that will convert the host genome into a oncogenic manner. Means before the virus attack, the host cell was not oncogenic. After the virus attack, upon incorporation of the viral genome with host genome, it is converted into a oncogenic transformation. It has undergone oncogenic transformation. Now the host genome will replicate rapidly, and it will cross the Hayflick limit of cell. It will not listen the barrier, it will not listen the contact inhibition, it will not listen to the tumor suppressor gene. Okay. That's why we are calling it oncogenic transformation. That is called V oncogenes, and C oncogenes means cellular oncogenes. C oncogenes means cellular oncogenes. Sometimes what happen, due to mutation of physical factor or chemical factor or some other factor, sometimes spontaneously, randomly, what happens, these cellular oncogenes are activated. The cellular oncogenes are activated, means it is called proto-oncogene to oncogenic transformation, randomly. There is no need of any chemical influence of chemical carcinogens or physical carcinogens or radiations. Spontaneously that can also happen. uh, that is called cellular oncogenic transformation. The cancer will start producing inside your body, start producing the colony of malignant tumors, and that will cause death ultimately. Okay, I think most of us, we are visiting cinema halls, and before a movie, you know, this person, this is Mukesh, they are tobacco consumers. So, how tobacco can cause cancer, throat cancer, oral cancer, they're the very good examples. So, please be aware of, be precautious, do not consume tobaccos, gutkhas, or bidis or cigarettes or something like that. Okay. Now, we'll try to differentiate between normal cell and cancerous cell. So, what are the properties you will find inside them? So, normal cells, cell growth is highly controlled and regulated, and obviously certain genes are responsible behind that. They are maintaining a proper limit of growth. Okay, what are those genes? Called proto-oncogenes. Proto-oncogenes are the genes responsible for controlling the growth and proliferation. Proliferation means cell division. I was telling Hayflick limit means, normally a Hayflick was a scientist, he said normally in our body a cell can divide 50 to 58 or 60 times, then automatically the set of this particular cell will die, a new set of cells gonna replace them. So this is how our body will maintain the total number of cells. Okay, you need to maintain the total number of cell of our body, and that is very much species specific. Different species has different Hayflick limit, and if you are failing to maintain the Hayflick limit, that means your proto-oncogenes are not working properly. Okay, with that we have some tumor suppressor genes. The P53 gene, that is responsible for, that inhibits proliferation, over proliferation. It is responsible for maintaining, if there is like your cells are proliferating upon the Hayflick limit, then your tumor suppressor gene will become activated, and the product of tumor suppressor gene will kill the excess cells. Okay, they are responsible for limiting the cellular growth. Okay, so both the genes are essential for cellular growth and cell division and proliferation, proto-oncogenes and tumor suppression genes. There are certain suicidal genes present into our body. They are responsible for the great phenomena called apoptosis or programmed cell death. Okay, how our cells, they will know that after how many divisions we should die. It's genetically programmed. After 50 division, the cells should die again, a new set of cell gonna replace them. So that is called programmed cell dead or apoptosis, in order to maintain this number of cells into our body. Okay, so these are some important genes which are present in normal cell: proto-oncogenes, tumor suppressor genes, and suicidal genes. Okay, normal cells have contact inhibition, means when the cells are dividing, suppose this is a single cell, when it will start division, it will maintain proper border to border contact. When the border is touching each other, then they will stop the cellular growth or cellular division. So that is called contact inhibition. It's just like that. There is a bench in your class, and in that particular bench only 4 students can sit, right? Now once there are 4 students sitting into that bench, the 5th student cannot be allowed to sit here, because that will create entropy or overcrowdedness. So that is called contact inhibition, right? When is following a particular limit, right? The 5th criteria for normal cells, you can observe the telomerase activity. After every division of cell, a special slice of DNA is cut out. After every division, basically, we are losing a certain amount of DNA. So that is due to the activity of an enzyme called telomerase. So telomerase activity is a predominant genetic activity or predominant enzymatic activity during the time of cell division in normal cells, and that is also responsible for the aging of us, and that is also responsible for the death of cells, right? Now this adhesions the cells can determine their boundary, cells can determine their volume and surface ratio. So all these are regulated by proper genes like proto-oncogene, tumor suppression genes and suicidal genes. Now we are moving forward towards the cancerous cell. What are the property of cancerous cell? Cancerous cell means uncontrolled growth of cells. That means the cells, they have lost the contact inhibition and adhesion property. They are ignoring the apoptosis, the program which is which is confining the cells and saying giving signal not to create overpopulation, overgrowth. Okay, so the addition property has lost, the contact inhibition property was lost, and the apoptosis property will also be lost. Now the cells have become immortal. The cells have become immortal. They will not die, they will not listen the Hayflick limit. So this condition we call it neoplasia. The cells will be called as neoplastic cells. They will start haphazard division. They won't be following additions, contact inhibition. Okay, there will be loss of cell addition, angiogenesis. In the newly growing cells, they will stimulate production of blood capillaries towards them. They produce angiogenetic factor. They're inviting blood supply because they need nutrition. Okay, and when they're inviting blood supply, they're exhausting the nutrients which you need to supply into your other healthy tissues or organs. Okay, metastasis means migration of the abnormal cells from one place to another place and establishing new tumor and new population of abnormal cells. That is called metastasis, the most fearful and most dangerous property of cancer. Okay, genomic instability will be there. They will suppress immune system, they will not listen to them, and the cells, this is the normal cell, it's a normal nucleus, but the cancerous cell, they have a large nucleus with abnormal chromosomes. Okay, so there you can differentiate between a benign tumor and malignant tumor. Now, meaning of benign is good, means kind of harmless. Malignant means bad or harmful. So tumor means extra like extra growth of tissue or unwanted cellular growth into our body. It is called tumor. That means the tumor can be growth outwards, the tumor can be grow inwards, right? But after a certain time, if the growth become confined within a certain region, okay, that started following the cellular addition rule. So that particular tumor is confined in a particular place and that is not harming you. So we will be calling that tumor as benign tumor. But if the tumor is not stopping the growth, that is that already had forgotten all the rules and regulation of normal cells, that is not following the cell addition rule, that is not following the contact inhibition rule, and the cells are getting transferred from one place to another place, that is the cells become neoplastic, the cells are migrating from one place to another place, means they have become metastatic. So we call this kind of tumors as malignant tumors. So with an example I am showing you, this is a normal human being, a tumor arises here, but that is confined only to this place. So we'll be calling this as benign tumor. But for another person, he or she observed a tumor here, but within few days the tumor is transferred another place. Within few days the tumor got transferred or migrated, forming a secondary population of tumor throughout the body. So this kind of tumors can be called as malignant tumor. So the difference is given here. Benign means kind, good. They are confined to their original location or the primary location. They do not spread to other parts. They will not show the property of metastasis. Okay, they will cause negligible damage or little damage. Okay, now when we talk about malignant tumor, malignant means bad or harmful, that has neoplastic cells, unlimited growth, grow very rapidly, invade and damage other tissues. That is called metastasis property, forms new tumors by displacement or by migration. Okay, and it is said that the most feared property of malignant tumor is metastasis. Okay, now we will talk about types of cancer. Okay, carcinoma, if there is cancer in epithelium, skin cancer, right? Skin is also one of the epithelial tissue, right? Carcinoma. Okay, sarcoma, cancer of mesodermal tissue like bone and cartilage, that is called sarcoma, cancer of mesodermal tissue. Sarcology means, or myology, sarcology means study of muscle. A muscle is originated from the embryonic mesoderm, right? So the mesodermal cancer is called sarcoma. Bone and cartilage, they are also mesodermally originated tissue. Okay, lymphoma and leukemia, they are cancer of blood. Basically they will attack lymphocytes or leukocytes, WBC. They are attacking WBC, that's called blood cancer. Okay, now the topic we are going to study, that is cancer detection and diagnosis. Okay, early detection is essential because uh, though the cancer is also a fatal disease, but there are few cancers if detected early. Remember, uh, there are few cancers if detected early can be completely curable, right? Though few blood cancers they are not completely curable, few bone marrow cancers they are not completely curable, and the treatment is also very much costly. So it said that few cancers like few breast cancer, prostate cancer, or skin cancer can be curable if detected in early stage. Okay. Now, how it can be detected or diagnosed? So first said that blood examination. Okay. So when you are examining blood, there is also pathological diagnostic test. Increased WBC count indicates leukemia or lymphoma, or detection of tumor marker. There are certain genes, there are certain genes present in malignant cells. When it's converted into oncogene, proto-oncogenes are converted into oncogene, p53 genes are switched off. So in that case those mutated gene will act as tumor markers. Okay, and those malignant mass of tumor will also release some proteins or chemicals. They will act as the product of those mutated gene. So we also can call those proteins as tumor markers. Okay, increased cell count will be there in case of leukemia. Okay, presence of alkaline phosphate into our bloodstream, or that is also example of that is indication of bone cancer. Increase alkaline phosphate, right? For bone formation you need osteoblast cells. If there is exaggerated number of osteoblast cell, there will be too much consumption uh, or release of alkaline phosphate will be there into the bloodstream. Okay. Now we'll talk about biopsy. Basically, what is biopsy? Histopathological study. So suspected tissue cut into thin sections in and stained and examined under microscope. Okay, with biopsy gun or a special kind of needle, surgical puncture will be there, and the biopsy gun will collect tissue sample either from the infected or the manifested tissue where you are suspecting cancer is there, either from the bone marrow or from the kidney or from the breast tumor lump, and in lab will study the histological aspect, different histological aspect of the cells present, and ultimately the pathologist will conclude that either there is abnormal cells are present or not. Okay, so that is called biopsy process. Okay, to detect cancer of internal organs. Now what about internal organs, visceral vital organs? With techniques like radiography, CT scan, and magnetic resonance imaging. And for radiography and CT scan, we use ionizing rays like X-rays. For MRI, we use non-ionizing rays. Okay. So computed tomography. So that is called as CT scan. So with the help of the CT scan, basically by utilizing the ionizing rays like X-ray, we create such kind of image like if you are finding colon cancer, that can be detected with the help of CT scan. Okay, this is how a CT scan is performed, right? Now the next thing is MRI, magnetic resonance imaging. MRI. A strong magnetic field and non-ionizing radiation is needed, right? A patient is inserted into that MRI machine, and outwards the body of patient, a strong magnetic field is rotating, and it's radiating non-ionizing radiation, and that helps us for accurate detection and pathological and physiological changes into the living tissue. Right here you can observe the development of cancer, how the MRI can form image where exactly the tissue is damaged or the cancer is manifested. Okay. Even you can use certain antibodies against cancer specific antigens. Okay. These cancerous cells or neoplastic cells, they will release some abnormal proteins. They can also act as a marker, and these proteins can be detected with the help of certain antibodies. Okay. Now, there are certain techniques of molecular biology. We can take help of biotechnology as well to determine the cancer. What are these techniques? PCR will help us to detect or to find the mutated gene, and autoradiography will help us to detect either either gene mutation is present into our body or not. So what happens for autoradiography? You need to take support of other techniques like PCR and southern blotting and the gel electrophoresis as well. So suppose this is the normal gel plate, southern blotted plate. So what we take here, we take some uh, primers which are tagged with radioactive molecules. What happened? We know the normal gene setup, normal genetic setup of our cell. So we'll take a cell, take few cells from our body. We'll take out the DNA from it and we'll give into the gel plate and we'll run the gel electrophoresis. We'll take the impression of those uh, DNA segments. We'll perform southern blotting, and what after that, after performing southern blotting, we need to incorporate some radioactive probes. These probes are supplementary, so not supplementary, these probes are complementary to the known DNA setup. These probes are complementary to the known DNA setup. Okay. So the process will be called hybridization. We'll take out the DNA, we'll form the, will denature the double strand, and the single stranded DNA are present. Now the probes will go and bind with the complementary DNA part. Now, when autoradiograph, when autoradiograph, you can detect the radiated portion by the help of the radio waves radiating from the successful bonding, which are produced upon successful bonding with the DNA chains with the radioactive probes. Now these are certain places where you will not find any impression of radiation. That means these are the mutated DNAs or mutated genes. If radioactive probes are not binding with the DNA, that means this DNA is already undergone through a process of mutation. So if there is no radiation is taking place from your plate, southern blot plate, that means obviously 100% there is presence of mutated gene and there is presence of cancerous cell into the body. Okay. Now, how to treatment cancer? The treatment of cancer, how to treat? It may be by the process of surgery, surgical removal of mass of tumor, right? It can be with the help of radiotherapy. It can be with the help of radiotherapy. It can be with the help of chemotherapy by utilizing some particular chemicals will targetedly will kill the neoplastic cells. Those are side effects are there for both the radiography and chemotherapy, but with advancing technology we are trying to minimize the side effect when we are performing radiotherapy and chemotherapy. Okay, there are certain immunotherapy. Now what happens in immunotherapy? Biological response modifiers or alpha interference are used against the cancerous cell, right? We know that cancerous cell, they're overriding our immune system. They're not listening. They're ignoring our immune threat. But with the help of this alpha interference, we call these biological response modifiers, we are detecting the cancerous cell and compelling them to follow the normal biological or physiological mechanism. Okay, this is how when someone is performing, when the doctor is performing radiotherapy, targetedly a particular uh, tissue is targeted and the rest of the tissue is unaffected, just covered with proper manner. Okay. So the next topic, the last topic of this chapter is going to be drug and alcohol abuses. Okay, so when you talk about drugs and alcohol abuses, we need to classify that what are the different types of common drugs which are taken, and obviously we'll be emphasizing on the abusing effect and the probable precautive measures which can be taken against the use of alcohol, against the abuse of alcohol, against the abuse of cigarette smoking and all. So drugs which are commonly abused, they belong to three group: opioid, cannabinoids and coca alkaloids. Okay, and majority of these drugs, they are obtained from flowering plants. Remember, different families of plant will be producing, flowering plant will be producing these different types of drugs, and few of the drugs are procured from fungi, right? Okay, so first we'll talk about opioids. Okay, they are extracted from latex of poppy plant. These are flowers of poppy plant, and if you make cut on their epidermis, it will start secreting the milky latex, and from this latex we are producing opioids or opioid drugs. Okay, uh, even in few areas the seeds of these plants or the poppy seeds are used as a ingredient of cooking food, right? but the latex is very much harmful for us. Okay, it can be used to prepare medicines as well, right? Okay, so the scientific name of poppy plant is Papaver somniferum. From latex we prepare morphine. Morphine is also used as a painkiller, okay, along with the narcotic effect, okay. And you can convert this morphine chemically into heroin. It's called acetylation. So this acetylated morphine is called heroin. The common name is smack. So morphine is the precursor form. After acetylation of morphine, you will get heroin, which is commonly known as smack. There is also has the name of diacetyl morphine. Now how it looks, so white in color, white powders, odorless, it has no smell, bitter in taste, and it will look like salt, so crystalline in nature, right? So this is how you can identify heroin or smack, okay. How it is taken? It is taken by snorting. This is how snorting is done, and also it can be taken as with injection, right? Now the effects you need to study. They act as depressant and slows down the body function, and that will give someone the temporary relaxed state, right? Mechanism of action, how it creates the depression or slow down the body function. It binds to specific opioid receptor. Now where these opioid receptors are present, these opioid receptors present on our central nervous system and gastrointestinal tract. So these opioid receptors are present on our CNS and GI tract. So these opioids, they will get inside our body, they will bind with these receptors, and they will cause this sedative effect, or that will gradually slow down the activity of our body, that will increase secretion of the inhibitory neurotransmitter, okay. That also effective sedative drugs or painkiller, sedative drugs or painkiller, the drugs which slow down our body activities, slow down the process of nerve impulse propagation, block the secretion, block the transmission of excitatory neurotransmitter, and stimulate the secretion and transmission of inhibitory neurotransmitter like GABA. Okay, so this is useful who have undergone surgery. So that's why the painkiller effect is though useful for us. Morphine, a potent painkiller, but overdose of morphine or use of heroin for long term can cause addiction, and ultimately that can cause damage of our nervous system as well. Okay, next, cannabinoids. Yes, I think most of us, we have seen this plant, right, somewhere. So cannabinoid drugs, they are obtained from inflorescence, means either flower or flower related part, the buds of flower, even from the leaves as well. Inflorescence of the plant called cannabis sativa, name of the plant, remember, cannabis sativa. We can use flower top, leaves, resins are also used with various combination to produce marijuana, hashish, charas and ganja. Like if you are preparing marijuana, there will be mixture of the resin of this particular plant with flower buds and leaf. For ganja, there will be mixture of the leaves with flower buds. For hashish, there will be mixture of resins with flowers. Okay. How they are taken by inhalation or oral ingestion. Ganja, charas, marijuana, they are taken by either you can inhale the smoke or also ingest with making a paste, when you can ingest with milk or any other fluid. Okay, it affects cardiovascular system. That means that increases your heart rate. Okay, how it will be affecting us, that interact with cannabinoid receptor. Now where these cannabinoid receptors are present, these receptors are present on brain. Okay, sometimes these are also used by some of the sports person, right? Okay, why some of the sports person they will use it? But because that can increase the heart rate, cardiovascular system, so that can kill the tension, or that can kill the stage fear, right? So when you are studying opioid drug, you must need to remember the structure of morphine and the plant called poppy plant or opium poppy. You need to identify this plant. You need to identify the structure of chemical structure of morphine. Okay, when you are studying cannabinoids, okay, you need to identify the leaf of cannabis sativa plant and the structure of cannabinoid molecule. Okay, this is the diagram of datura plant. Okay, datura also a flowering plant which we use uh, for like puja purpose of Lord Shiva, right? Okay, coca, next type is cocaine or coca alkaloid. Coca alkaloid or cocaine. They are obtained from coca plant. Do not confuse with cocoa plant. That is different. From cocoa we get chocolate. From coca we get coca alkaloids. Scientific name is Erythroxylum coca. That is a native plant of Southern America. So we commonly call it coke or crack. Remember the common name of heroin, that is smack. Common name of cocaine or coca alkaloid, coke or crack. That is usually snorted, right? Usually snorted, that interacts with transport of the neurotransmission dopamine. That's why few athletes also can use this to kill the stage fear. See the dopamine, that is called happiness hormone. Okay, so uh, interacts with transport of neurotransmitter dopamine. It inhibits the reuptake of dopamine. So the more the dopamine present into the synaptic cleft, it gonna stimulate the postsynaptic neuron, and if the postsynaptic neuron is getting stimulated, that means that will create hallucinogenic property into your body. You will laugh, you will feel energetic, you will feel that you are happy from inside. Okay, so mode of action, so it stimulate CNS, central nervous system, produces sense of euphoria, that is happiness, and increased energy. But excessive dose of cocaine can also cause hallucination. Hallucination means, suppose you have taken excessive dose of cocaine. Now I will show a rope, and I will, by showing this rope, I will tell, see, this is a venomous snake. So what you will see, you will actually observe, there is not a rope, that is a cobra. but actually that's a rope. But you are mistakenly, when you are into hallucination, you will observe it that there is not a rope, that is a cobra inside your imagination. So that is called hallucination. Sometimes hallucination occurs in some person that they can observe a ghost or spirits around them. So that is also kind of hallucination. Okay. Some other plants with hallucinogenic properties: Atropa belladonna, this is the plant, and Datura, this is the flower of Datura which is offered to Lord Shiva, and also a drug is obtained from this Datura called Daturine that has this hallucinogenic property. Okay. Barbiturates, amphetamines, and benzodiazepines are normally used as medicines to help patient to cope with mental illness like depression or insomnia. Insomnia means if you are unable to sleep, that condition is called insomnia. That means you are under too much stress or tension. So you can be treated by giving or administrating this kind of drugs, barbiturates or amphetamines, but these are often abused. The overdose of barbiturates or amphetamines can cause sedative effects or even hallucinogenic property they have. Smoking also paves the way to hard drugs. So basically what happens at the age of adolescence, uh, by observing your friends, by due to the peer pressure, few of the adolescents, few of the teenage, they start smoking. Okay, and gradually from smoking to alcohol to heroin, charas, ganja, and that will be paving pathway towards other hard drugs, and ultimately the future is dark. So avoid all these things. So tobacco has been used by human for more than 400 years. A statement can be given. Statement can be given. Use of tobacco is longer than 400 years. So how tobacco is used? In the form of biri or cigarettes, it can be smoked. In the form of gutka, it can be chewed. In the form of this brown sugars, it can be snuff. Okay. Tobacco contains large number of chemicals including nicotine and alkaloids and few other aromatic components. Okay. Now the function of nicotine on our nervous system. The nicotine stimulates the adrenal gland. Ultimately, adrenal gland is under a sympathetic nervous system to release adrenaline and noradrenaline, and the effect is raise in blood pressure and increased heart rate. The effect of adrenaline and noradrenaline on our cardiovascular system. Adrenaline and noradrenaline, that they're also known as epinephrine and norepinephrine, they're also known as catecholamines. They will increase our heartbeat and heart rate, stroke volume, right? So increase blood pressure. Okay. Cancer of lungs, urinary bladder, throat or oral cavity can be caused by chewing the tobacco. You know that tobacco contains PAH, many other aromatic components. They're responsible for cancer. Smoking can also cause bronchitis. The aerosols or the smoke particles will cause inflammation of our bronchi and bronchus. Emphysema, damage of alveoli or respiratory surface, and coronary heart disease, deposition of minerals in the wall of a coronary artery, and obviously deposition of lipids and cholesterol. Okay. Gastric ulcer can be caused. So smoking increases carbon monoxide content of blood, and you know that if there is even 1 or 2% carbon monoxide content, carbon monoxide is present into your body, that will directly oxidize the hemoglobin into carboxyhemoglobin, and that may kill you. Okay. So these are some examples how tobacco is marketed in cigarettes, Bimal, you know, right? Okay. Uh, though the ad is not of a tobacco, the ad is of elaichi, but few other vendors of Bimals I have seen that they are selling uh, tobacco, and if you are consuming them, it can cause oral cancer or throat cancer or even other diseases. Okay. So regular use of alcohol can cause damages of nervous system and liver cirrhosis or fatty liver. So fatty liver, if you are consuming alcohol, the detoxification is happening inside our liver. Liver is our detoxifier of fire. But if you are providing overload to your liver, then what will happen? Gradually, the liver will start converting the alcohol into fats. The excess fat will accumulate outside the liver, and the hepatocytes will lose the reactivity. Okay, and the physiological functions of the liver gradually will decrease, and ultimately the liver will start dying. That is called liver cirrhosis. Okay. Now, if somebody is a chain smoker or a consumer of alcohol or other drug, now if you say suddenly that please stop consuming all these things, this means withdraw of alcohols or withdraw of drugs. So here she will face some symptoms of addiction withdrawal. Okay. So what are those withdrawal symptoms? Nausea, the feeling of vomiting, shakiness, okay, change of mood fluctuation, aches or pain, headache, stomachache, hallucinations, sudden perspiration or sweating, loss of sleep, depression or anxiety. But these are temporary. There is a proper methodology for if you are going for withdrawal of a drug or smoking, or if you want to get rid of alcohol, gradually you have to like give stimulation towards your nervous system. Gradually you have to stop utilizing, stop consuming all these things. And here the things are not biological mainly, things are mainly mental, psychological. So psychologically you have to build yourself more and more stronger, right? You indulge yourself to some constructive works, and that will really work. Okay, now let's solve this question. So which of the following statement is correct about tumors? Okay, tumors, we know there are two types of tumors, benign and malignant. So it said that benign tumors normally remains confined to their original location. Yes, do not spread to other parts of the body and cause little damage. That is absolutely true fact. Okay. Second statement is saying the malignant tumors are mass of proliferating cells called neoplastic tumors. Absolutely correct. And these cells grow very rapidly. This is also true. So, if you are searching for the correct statements, both 1 and 2, they are correct. Okay. Next question. Which is the particular type of drug that is obtained from the plant whose one flowering branch is shown? So basically that is branch of Datura. Okay. So by from this plant you can obtain hallucinogenic drugs. We were studying here only, right? Remember? plants with hallucinogenic properties, Atropa belladonna and Datura. So, with this, I am concluding uh, the lecture of human health and disease. We will meet in some other class. Till then, Jai Hind, bye bye.